Thank you for coming to this industry panel. I was just telling the panelists or the speakers that in the past, of course, we're in the Twin Cities, so we're fortunate to have a lot of medical device ecosystem here, especially. Oh, come on up. Do we have a seat for you there? Yeah. And we have, fortunate to have a lot of neuromodulation or neurotech companies locally, but also they have reach out in the U.S. and the world. And we've done panel discussions in the past. Those have gone well, but there's always cool things going on by the companies that we don't fully get into. So we thought this year, how about we have them come to the conference and actually share a lot of the cool developments and technologies and research that's going on in each of their companies. So that's what we decided to do. And if there's a little bit of time left, we'll have questions at the end. But more, this was just kind of informative to learn about these different companies. So I will, we have 7. Oh, they're all listed. Nope, not there. Okay, well, I'll just introduce. I'm not going to introduce each of them until they speak, but we basically have Dr. Jeff Kramer from Medtronic, Dr. Kent Lee from Inspire Medical Systems, Dr. David Page from Abbott. Maybe, actually, I should have had you raise your hand just briefly. There, Dr. Kramer was first. Yeah, and Dr. Kent, thank you. We also have Dr. Andy Kelly, and then Dr. Mark Hjelle from Heraeus. Sorry, Andy Kelly from Cirtec Medical, and Dr. Mark Hjelle from Heraeus Medical Components. And then we have Dr. Gaynor Foster from Maxtim. And then finally, Dr. Merritt Gorley from Boston Scientific Neuromodulation. Okay, so what we'll do is we'll have them come up and give their presentation at the podium. And the first speaker that we're going to have is Dr. Jeff Kramer from Medtronic Neuromodulation. He's the Vice President of Research and Technology there. He has over 20 years of experience in neuromodulation research across a number of therapy areas. He has worked and consulted for a number of different successful startups and larger device manufacturers, leading research and clinical teams to help bring multiple therapies to market. He has a PhD in integrative physiology, focusing on neuroscience from the University of Illinois. So thank you for taking the time to give this presentation called Biosensing in Neuromodulation Therapies.
Here we go. Hello? Can you hear me? Wonderful. Well, thanks everyone for attending this great symposium, and thanks to the hosts and organizers for it. It's always a great annual event, and Medtronic's really pleased to be able to sponsor this. I think we have 7 minutes of fame up here, something like that. So this is going to be kind of a whirlwind review of. I thought what we'd do is talk about some of the biosensing work that Medtronic's been doing now for quite some time. I'll just say up front that all the credit for this goes to all the Medtronic research and technology team members who are here. There are quite a number of them. It's in our backyard, so we like to be able to attend. And so some of the work that you'll be seeing here is either done by them or is continuing to be done by them. So just props to the team here on that. This is a disclaimer. This is all research and technology, so not necessarily commercial elements. I think you're all aware of that. Now, I'm a systems physiologist, so I would be remiss if I didn't talk about Claude Bernard, for those of you who are historical buffs for physiology. So, as you probably know, Claude really developed the whole concept of homeostasis, which was a term that was coined by another individual later on in the 30s. And really the whole basis of this is that there's a signal of some kind that's sensed by the body, and that signal is then used as a marker or as a physiologic indicator that a change in function is required. So this is sort of the basis of closed loop physiology in a way. So it's how the body is regulating physiologic function. And so utilizing this as sort of inspiration for some of the closed loop therapies that we've been designing that many of you know about, where we work with a number of you in the room on some of this, that's kind of in my mind, the physiologic correlate. The technology basis for this, for us, is really on developing technology platforms that we can unlock to then further study these effects. So we've been doing this in the brain, We're doing it in the spinal cord now. We'll talk a little bit about that. But this is really the technology underpinning for us to be able to quickly advance these therapies, not just for research purposes, but eventually for commercial purposes, which is an important translational element for us. So we've been taking a lot of time to think about the technology overachievement, if you will, and then being able to have that increased capability with software unlocks or firmware unlocks. So this is something that, from a technology standpoint, we feel is really important. Many of you know more about this than I do in the audience, but in terms of signals or biosignals, there are many different kinds of biosignals. Of course, we're interested in biosignals in the central nervous system, in the brain and spinal cord. And this is just an example of some of that work from the STN and looking at these biosignals that you can measure and then utilize as a means to control stimulation or control therapy. So if we look at some of the classic work that's been done, and again, many of you are familiar with this, looking at the LFP signals on the power spectrum, looking at some of the frequency bands like beta, using a beta signal potentially is a way that we can then control therapy and tailor it and personalize it and gain therapeutic benefit from it with DBS. So the current capabilities of our system, Percept, is one that we can actually send signals. And now we're doing the clinical work to validate using beta in a closed-loop phenomenon or closed-loop therapy approach. But there's also future uses, of course, for neural sensing on top of closed-loop. And this is some of the work that's ongoing right now. So this is looking at sensing as a means to really facilitate and make programming easier for physicians and for patients. So being able to use the signals to custom tailor the therapy is but one indication or one means to utilizing those biosignals, but increasing the speed and ease of the programming and ease of the use of the therapy is another area that we're involved in. And I think that there's a lot of other areas that we're not going to have time to talk about, but this is one of those sort of near-term areas that we're doing some interesting work on. So now if we shift from the brain to the spinal cord, interestingly, one of the fundamental elements of spinal cord stimulation that we really still don't have a great handle on is dosing. How do we adequately and optimally dose spinal cord stimulation? And so if you look on the left, there's an amazing amount of variability of electrical dosing that the spinal cord sees in general everyday activities, daily activities of living. And really what we want to get to is what's shown on the right. This is a dosing paradigm that's much more consistent, that's maintained within a therapeutic window, and so you can avoid any side effects but maximally deliver the therapy. And so one of the ways that we potentially can do this is by listening to the nervous system. So my grandmother had a phrase, you should be listening twice as much as you talk because you have 2 ears and 1 mouth. So my grandmother was pretty smart. So what we want to do is listen to the signals that the spinal cord is giving us when we stimulate it. So we can ping the spinal cord, record an ECAP or an evoked compound action potential, so somewhat different than what we're doing in the brain. But then that ECAP signal, which is shown on the lower left here, is a direct indicator of the amount of dose that's being delivered to the spinal cord. So we can use this as a feedback control system. And we're able to record these signals, these ECAP signals, 40 to 60 times per second. So we've had more than 200 patients worth of data now on this. And we can reliably record these ECAPs and use this to control stimulation output. Now, what's interesting about the ECAP signal, of course, is that it encompasses much more than just dose. So what's encapsulated in the ECAP signal from a data standpoint is broader than just the dose. And that's another avenue that we're starting to explore now. This is an example of some human data that we've collected. On the left is an open-loop spinal cord stimulation, so what we traditionally deliver now. And what's on the right is a closed-loop using ECAP control. And what you'll see is that the tightness of the ECAPs, the amplitude variability is much less with closed-loop, which is exactly what we would expect. So when the variability of the ECAP is decreased, the dosing is more consistent across the time period, and what you'll see on the bottom here is the amplitude changes of the delivered voltage. So this is basically an ECAP-controlled closed-loop system for spinal cord stimulation, and basically showing that in humans, we can rapidly deliver much more consistent therapy and dosing to the spinal cord. So in general, I know that was a big whirlwind of biosensing. We're interested in biosignals, not just from the central nervous system as well. We're recording biosignals from the pelvic floor. We're recording biosignals from other peripheral nerves. And so this is a means for us to be able to control therapy in a much more patient-centric, patient-tailored way to hopefully be able to address some of the clinical issues that we see with lack of consistency of dose, both temporally as well as from a patient-to-patient perspective. So this is I think we're in a new revolution of neuromodulation. I think we're in a new technology curve when it comes to the actual devices that we're creating now with neural sensing capabilities, and we're really excited about the future for this. So thank you for your attention.
Thank you, Dr. Kramer. It was perfect timing. So great. Thank you. Next speaker is going to be Mr. Kent Lee. He's a research fellow and at Inspire Medical Systems in Golden Valley, Minnesota. He brings over 20 years of global experience in the implanted device and sleep apnea diagnostic and therapy sector, having developed the apnea scan diagnostic while at Boston Scientific, CRM, and while at Inspire, ran the EU arm of the STAR FDA approval trial and developed evidence for key peer-reviewed publications that led to Inspire's FDA approval and reimbursement coverage. He received a bachelor's and master's in biomedical and electrical engineering from Case Western Reserve University and an MBA, I didn't know that, MBA from INSEAD, and is an author of over 40 patents. So welcome and looking forward to your talk.
All right, thanks for the introduction. So hi everyone, my name is Kent Lee from Inspire. I lead our research efforts there, and the goal of my presentation today is kind of talk about an example of how you take a new therapy like Inspire or hypoglossal nerve stimulation for sleep apnea from kind of a concept into clinical phase. A little bit about myself. So I'm a biomedical engineer by training from Case Western Reserve University. I've kind of had two phases of my career. I think the first half I actually was at Boston Scientific, so a large medical device company. And then 11 years ago, actually, I joined Inspire. So at the time, which was a very small startup, just getting ready to start their pivotal trial for sleep apnea. And now here we are, kind of a therapy stage company, having treated over 20,000 patients within the past 5 years. So I'm going to do a quick introduction about sleep apnea, and then we can go into some of the really interesting details about how do you take a therapy to market. So if you're not familiar with sleep apnea, it's probably one of the most common comorbidities in middle-aged adults. So it's approximately 20 million people have sleep apnea. And what happens during sleep apnea is the muscles of the airway relax, and the most potent muscle is actually the genioglossus, or your tongue. What happens is, as you sleep, your kind of tongue relaxes, and in some people it causes closure of the airway. This closure of the airway then kind of leads to oxygen desaturation. You then arouse. From that, you wake up. Essentially, you hyperventilate, but then as you go back into sleep, you go back into these periods of apnea. This is bad because if you can imagine this happening, 15, 30 times per hour, that's like an alarm ringing 50 or 30 times an hour during your sleep, and most patients are very tired. There's a really great effective therapy out there right now. It's CPAP. I don't know if some of you may be familiar, might have family or friends that use it. It relies on a mask that sends pressurized air into the airway to keep the airway open. It's super effective. It's been around essentially since the 1980s. But one of the biggest challenges with CPAP is that only half the patients can use it. As in, everyone can use it, but only half of the patients are able to actually adhere and continue to use the therapy. And so that means there are people out there who essentially have sleep apnea but are undertreated or untreated. And so looking at the spectrum of therapies that are out there, CPAP is kind of the standard of care. It's the first choice. And depending on your severity of sleep apnea, there are other options. In the mild case, there are kind of very simple behavioral options that you can take. Or oral appliances. It's something from a dentist that helps to move the jaw forward. It's like a bite block, if you will. And so at Inspire, in the early days when we saw there's just a huge therapy gap. You had patients that had moderate to severe sleep apnea. They couldn't use CPAP. At the time, there was another surgical therapy, which is maxillomandibular advancement. So you simply break the jaw and the maxilla, and you move that forward. As you can imagine, that's not very popular. It's very effective, but you essentially can't eat for a month. And so there's this therapy gap there. And so we thought, can we utilize some science to create a new therapy that addresses this therapy gap? And that's exactly where Inspire was placed. So first, I think it's really interesting for us to think about when you develop a new therapy, you need to have some kind of mechanism that you work on. And so, as I mentioned before, the tongue relaxes, and that kind of causes the airway collapse. And we are really fortunate in the 80s, essentially, or early 90s here. This is actually also a case. There were physician scientists trying to understand what is the cause of sleep apnea. And on the tracing, you can see here, they had placed chin EMG on the genioglossus, which is right underneath your chin here, and they saw two things. They saw, one, the EMG decreases during an apnea, so there's something about muscle tone that's related. And two, they see that this muscle tone is synchronous with respiration. So as you breathe, actually your genioglossus activates a few hundred milliseconds before you breathe to keep the airway open. And so, obvious question, I think, is can you modulate this? And there was early work that was done. I think the first approach, which is not seen here, is a group in Japan, which they tried to put chin electrodes, so surface electrodes, and stimulate the genioglossus through the skin to see if they could reduce the AHI, the severity of sleep apnea. And the first thought, it works. They saw that there's a decrease of the sleep apnea. But if you drill deeper into the details, it wasn't that it was actually working. It was actually waking the person up. And when you're awake, you don't have sleep apnea. And so it's like you have this context, but that approach didn't work. So then, the next question, then, this is where these physicians started to work with Medtronic, is can we go after the muscle? So, the next phase is fine wire electrode placed directly through the mouth into the muscle, and you start to activate that. And actually, there was a sign. There's a sign you could open the airway. There's a sign that you could do it during sleep, and it wouldn't wake up the person. But it's not a conceivable therapy. You have electrodes sticking out of your mouth. So by the 90s, then, the next obvious question was, can we take a cuff electrode, place it around the nerve that controls the muscle, and then that's where we started to see, if you look at the figure in the middle here, each of these pulses are the pulses from stimulation. When you omit a stim burst, you see the arrow below, this is airflow decreases. So now you're on your way to a therapy. You can stimulate the nerve with a cuff electrode. But we learned a ton. There's a lot you have to think about, where on the nerve do you place the electrode? There's this granular detail you really have to go into. And that's where Inspire had done a lot of work to essentially develop a system where we found exactly the spot we wanted to stimulate. We needed a closed-loop system because we knew we had to be gated with respiration. But then you also need the externals. You need a physician programmer to program it. You need a patient remote, a remote to help the patient, titrate the therapy or turn it on and off at home. And essentially what we've built most recently then is kind of a patient management platform, a way for the physicians to remotely get information about the trajectory and the adherence to the therapy. Check my time here to make sure I'm still on time. I'm going to show you kind of a diagram that when you understand the mechanism, you can actually see how things are, how the therapy works. So we'll see if these play here. Having some issue with the animation, but you can almost tell here where on the left, this is actually the airway unstimulated, and this is the base of the tongue. You can see that's closing off there. Those bubbles aren't moving. And then on the right, this is at the soft palate that no air is moving. But once you stimulate, let's see if we can get this to run here. So every movement of the tongue that you see here, this is actually stimulation running to the hypoglossal nerve. You can see both the tongue base and the palate opens, and now you have kind of, you've relieved the apnea. We essentially took this to trials here, and I'll show you what this looks like, where as the patient breathes in, you can see the stimulation burst running, and there's just mild protrusion of the tongue in that video there without waking up the patient. So we took this therapy essentially to the pivotal trial in 2014. That's when I joined Inspire. And we were able to really demonstrate significant reductions in sleep apnea severity, essentially back down to normal levels, both in the objective and subjective measurements. And this really confirmed all the science that we were able to leverage to essentially deliver a therapy to market. So I'll say that in the path of delivering new therapy, the science and engineering is one, but a large piece of skill that you need to have. So you need to have reimbursement. That's essential for us afterwards. You need to have regulatory, tech development, operations, and so I guess for science and engineering people, we really focus on this piece, but it's being able to work with this entire team to take a therapy market really makes a gigantic difference. And with that, I will end the talk, and I'll just say we're hiring. So we are hiring scientists and engineers, and so we are a first mover in sleep apnea. It's a super exciting space. People really get what does it mean to have restored sleep, and so I think we have a booth out here. If you want to grab me afterwards, we'd love to chat with you all. Thank you.
Thank you, Kent, for your nice talk. And to my grad students, you need to finish your grad program first before going to Inspire. Okay, so the next speaker is Dr. David Page, staff research scientist, applied research at Abbott Neuromodulation. Has a passion for driving product design from research through product development and onto market launch. He has a background in clinical trial design, market and product needs assessment, preclinical studies, and product development, with specific background developing novel techniques in neuroengineering, sensing, and machine learning. Measures his impact by his ability to bring the new technologies to people that need them in a way that makes their life better. He's driven to identify technologies that improve quality of life for people worldwide. Prior to Abbott, he held roles of increasing responsibility at Avanos Medical, obtained his PhD in bioengineering from University of Utah, and a bachelor's in mathematics from Brigham Young University. So looking forward to your talk titled Patient-Centric Breakthroughs to Fuel Neuromodulation. Thank you.
Thank you, Hubert. Can you hear me? All right. I feel really privileged to be able to represent Abbott here today. I'm representing a big group of people who have collaborated on this work. Erica Ross leads our team, and a really capable group of scientists, engineers who deserve the credit for this. Obviously, I'm an employee of Abbott. That's why I'm here. So I want to talk about the concept of engineering scale and perspective here. And this is a graphic I pulled off of mathisfun.com because math is fun. And on the far left, you see the estimated diameter of a quark, 10 to the negative 8 meters. And on the far right, the estimated diameter of the observable universe, 10 to the 27 meters. And where we live is somewhere in the middle. And we tend to kind of focus on the scale of things at our fingertips, the size of our hands, or if you're a biologist, maybe the size of what's in the microscope where you spend all your time every day. But it's really important to step outside of that context and consider the context of what's going on around us on different scales. And for neuromodulation, that's absolutely true, and that's kind of what I want to focus on today in two senses. Number one, the concept of focusing on patient-specific factors, the person as a being, and the adoption of neuromodulation for patients, and then also people working together to bring the technologies to patients. And this is all kind of in the way of Abbott seeing ourselves as positioning ourselves as being a leader in digital health, which is really taking healthcare by storm. And so, in neuromodulation, we tend to focus a lot on implanted electronics and IPGs and leads and all these things. But it's good to look out and realize that a lot of other disease states and diagnosis are now being managed in different ways that are more seamless, and we're moving neuromodulation in that direction. So I want everybody to take off your science heads for just a minute and think about a person who you know that could benefit from a neuromodulation therapy. Goodness. Do I need to plug this in? Yeah. I'll try to pretend like I knew what I was doing. I like two hands. It's okay. Thank you. Somebody has to. Let's slide it over so I don't tap the cord with my elbow again. we go. All right. I get 2 rounds of applause. I feel really privileged. So think about somebody you know who could benefit from a neuromodulation therapy or somebody who does. And now kind of distance yourself from the therapy itself and think about that person's life. And what is it that they care about on Thursday, Friday, Saturday, Sunday, and every day of the week? What are the things that really matter to them? And how does the neuromodulation enable them to be involved in the things that matter and not take them away from it? And when you're a patient, you're deciding whether to get an implant, you really have to consider cost benefit of, what's this implant going to bring to me, but what is it going to take away from me as well? And neuromodulation in the past, I think, has been seen by a lot of patients as it comes with trade-offs, right? One that's really well known is MRI compatibility. A lot of patients just wouldn't get an implant before because it wasn't MRI compatible, and you're probably going to need an MRI at some point. And we're overcoming that with a lot of innovations in that space. But something new that we've introduced in the last year is breaking this geographic barrier. So the travel barrier. If somebody who lives 5 hours away from a major clinical center, can they really handle driving 5 hours to be reprogrammed and then drive 5 hours back home every time they need to be reprogrammed? And so we introduced Virtual Clinic, which is more than just a video chat with your doctor. It's a video chat, but your doctor can program your stimulator theoretically from the other side of the country through the internet. And that involves a lot of innovation in cybersecurity. And you imagine being able to program an implanted device through the internet. We got designation by Time as one of the best innovations of 2021 for that technology. So really exciting stuff. Now thinking a little bit about the interaction of the device with all the different people involved in bringing a patient to the therapy. So here's a depiction of what a patient goes through from when they identify neuromodulation as a possibility to when they actually get the implant and live with it. And I think the point of this is that there are a lot of steps and it's a long, tortuous pathway that's not well managed. And that is a barrier to a lot of patients as well. And so digital health and digital technologies can come in to help resolve some of that and streamline the process for patients. I'm going to skip through some of this. We have kind of a digital ecosystem we've built out where patients bring their own device. They can control their stimulator from their own smartphone. We collect surveys from them on a daily basis from their smart devices. But really where we're going next is what we refer to as seamless data. It revolves around the idea of giving the patients their device and allowing them to forget about it. Forget about their disease state, forget about the device that they're being treated with, and just go live their life. And seamless data comes with a bunch of other, positive characteristics like being objective, not relying on survey questions that can be hugely biased and affected by, the events of the morning and the environment where you're being asked, and being multi-parametric, so starting to look at the effects of neuromodulation devices, not only for pain, movement disorders, but also how does it impact the patient's sleep, their mobility, their mood, things that actually really matter to people and have a tangible impact on their daily life. And then being timely, and this is very important because right now, a physician, they get this kind of snapshot when the patient comes in of this is how I feel right now, and 2 months later the patient comes in and that's how they feel right now. But you don't see this continuous portrait of what happened in between. And so we're building in our digital infrastructure to create that portrait for the physician when they go in for their visits with the doctor. This is a clinical study we're running right now called SCS Trial to Perm, where we're monitoring patients before the trial period for spinal cord stimulation, during the trial period, after the trial period, and after the implant. It's a longitudinal study where we're monitoring patients both with commercially available sensors like what you see here, things that probably a lot of you are wearing today, and then also a validated chest ECG sensor that is completely wireless and battery powered. We collect sensor data throughout this entire period from the patients. We collect daily surveys from them. The idea is to use the sensor data to predict what they would have said on the surveys so we can scrap the surveys eventually and have this kind of seamless experience where we know what's happening with the patients, but we don't have to ask them. And some of the interesting sources of data that we found to be important, in addition to all the wearable data, are interesting things like weather and seasonality. Turns out in our data sets, it's a great predictor of pain on a daily basis. Demographic information, and medical history, all of these contribute to creating this kind of picture of what's going on with a patient and then predicting what's going to happen, and building kind of a personalized therapy for them. This is from another clinical study that we ran called the Reality Sub-Study. On the left-hand side, you'll see a bunch of features that we collected. These include things from wearable devices, medical history, and essentially we put all of these features into a machine learning algorithm and used them to predict whether a patient had mild, moderate, or severe pain on a given day. And with that data set, we were able to predict what their pain level was with an accuracy of 90%, simply based off of these seamless data sources using a random forest model. So really exciting work, and we're advancing that forward. And as I'm kind of closing up here, I just want to point out the keystones of the digital health transformation that is kind of taking healthcare by storm. The concepts are the Internet of Things, putting sensors in everything and in everybody, or on everybody. Number 2, artificial intelligence and automation, machine learning. And then number 3, big data, and being able to handle a large volume, velocity, and variety of big data, including data that may not be really clean and cut and dried, but being able to handle missing data on the fly. And incorporating these aspects into commercially available implantable devices into the whole system in a corporate environment is not simple. It takes a lot of work and a very innovative mindset, but we're doing it, and it's really exciting to see what's coming of it. And it has really opened our minds to not only more data, but data that will lead us to more truth. And so as I invite you to kind of expand your perspective, to think a little bit about in your studies, like what other data sources you can look at or add in to broaden your horizons, not only about what solutions you're looking into, but probably more importantly, to help you understand which questions to answer, because that ultimately leads us to where we want to go. And so I'll end with this slide. Without big data, you're blind and deaf in the middle of a freeway. And if that wasn't true 20 years ago, it absolutely is true now. So thank you for your attention.
Thank you, David. Great. So the next speaker will be Mr. Andy Hjelle from Cirtec Medical. Andy Hjelle is the Director of Applications Engineering at Cirtec Medical, formerly Cactus Semiconductor, located in Arizona. Prior to joining Cactus Semiconductor, he was Senior Principal IC Design Engineer at the Medtronic Microelectronics Center. Throughout his 30-plus year career, he has defined and designed more than 30 full custom mixed-signal ICs for a wide range of portable, wearable, implantable, and ingestible medical devices, such as glucose meters, hearing aids, my field, neurostimulators, cardiac pacemakers, drug infusion devices, biosensors, orthopedic sensors, and ingestible sensors. He is a senior member of IEEE and serves as workshop committee chair for the Phoenix chapter of the Engineering and Medicine Biology Society. He also serves as chairman of the Industry Advisory Board at the Center for Neurotechnology. Today he'll speak about Cirtec Medical, recent innovations for neurological sensing and closed-loop neuromodulation. So thank you for providing your talk.
Thank you. And is this working? Oh, now I hear. Yeah, and thanks for the introduction, and thanks very much to the organizers. This is a great opportunity, and we appreciate it very much. I don't think people know as much about Cirtec as they know about some of the big OEMs we talked about, so I'm going to start a little bit with just an overview of who we are, but the focus is I'm going to get into some of the new things we've been working on. So, high level, we were founded a long time ago in 1987. We are essentially a development and manufacturing, contract development and manufacturing, not exclusively, but almost exclusively for medical devices. We have designed and thank you. Yeah, we have design and manufacturing operations in several states across the country and several countries around the world. We have about 4,000 square feet of manufacturing and office space and over 1,000 employees now. So we've been growing quite rapidly over the last 4 or 5 years. We have a strategic focus in Class 2 and Class 3 medical devices. We do, the highlighted one there is an awful lot of neuromodulation and active implant systems. We also do cardiac and drug delivery, circulatory support. The list goes on. We participate in a lot of these devices at a lot of different levels. And we also make the components that go in them. So in some cases, we can make the overall enclosure or an entire device. And in some cases, we can just add the one missing piece to your puzzle that you need to manufacture your device, but always on a contract manufacturing and development basis. We've developed a Neuromodulation Center of Excellence here in town, up in Brooklyn Park, where we are operating around a platform of devices. So we've got a couple devices like the picture shown there, full IPG with patient programmer charger, cloud control, the whole picture. So we have a couple of these that are available for use as basically as a proof of concept device to get you started on a new therapy or on a new device and potentially customizable as you get further down the line. We also have the guts that go in those things, the electronics. The group I work on is ASIC development and production. We have thin film flex circuits. We do the leads. We do the patient programmers, et cetera, and manage the whole supply chain. So the idea is basically a one-stop shop. If you need all of our services, you come in the front and get the device out the back door. That's the plan. So most of our chip business where I work is full custom, but we have a couple what we call application-specific standard products, which is a bit of an oxymoron, right? A specific custom device. But anyway, that's the name we use. So we have a couple of those in the neurostimulator IC area. I won't go through the details, but there's a variety. There's one with a real high channel count, one with a real small channel count. We've also got power management ICs, and for several of these we have development kits. So you can buy a couple prototypes, you can buy the development kit, you can essentially build your device on a breadboard and make sure you can do everything you think you need to do, and then once that's done, then you can move forward making it into the right form factor. So what's new? We've announced this CSI 080. It's a 4-channel neurosensing IC. So it's going to be another ASSP like we mentioned on the previous page, but instead of sensing or power management, instead of stimulation or power management, this one's for sensing. So some of the details, it's based on a patented pseudo-random chopper amplifier. So it's getting kind of EE-ish here, but anyway, the basic advantages of the input, chopping the input of an op-amp eliminates what we call 1/F noise. So the basic fundamental resistors and transistors that we use in our circuits are noisy, right? So they do not always produce the same result, which looks like noise in your output. One of the primary drivers of that noise is what we call 1/F, and it's inversely proportional to the frequency. So while these EEG signals and the local field potentials that we measure, they're super low signal bandwidth, which is first you think is good, but they're so low that it's actually bad. Because if you think of 1/F, and if 1/F is really low, you've got a really high noise floor. So the way you get around that basically is with a chopper amplifier where you sample a signal and then you sample the inverse of that signal and you chop back and forth and recreate it. So the chopping eliminates the 1/F noise. We have a closed loop architecture that enables us to do that all with ultra low power. Traditional old school chopper amplifiers, if you can think about it, if you're looking at the signal and you're going to add a high gain, the output of your amplifier has to slew all the way to the signal plus and then all the way back to the signal minus and then back and forth, thousands of times higher rate than your actual signal bandwidth. So it burns a lot of power. With the closed loop architecture, it doesn't actually have to do that. You're sort of modulating the signals and the impedances inside, so it enables much lower power. In this architecture, capacitor ratios set the forward gain. So a lot of times when you're doing a forward low noise amplifier, the gain is set by a single component, and so the variation in that component leads to variation in your gain. When you're using a ratio of components in our IC world, things match very well. So the ratio is a very, very well controlled signal while the absolute value is not. So in this case, the feedback cap set that gain, so it's highly repetitive and highly repeatable. Those same capacitor ratios set the frequency bandwidths, right? So when we set a frequency bandwidth of 1 hertz, it's really at 1 hertz. It's not plus or minus 20%. And then the patent part is this pseudo-random frequency approach. And so the idea here is we talked about chopping back and forth on that input signal, and simplistically you would do that at the same rate at all times. But that chopping frequency now becomes a noise source. right? And so you're trying to get rid of noise, and the first thing you do is introducing noise, right? So this patent now says we're going to essentially spread the spectrum around that. So in a pseudo-random fashion, we're wiggling around the frequency of that, so then we can more effectively filter it out, and the net result is a lower noise. And so that's the patent. This is an advancement on previous work by lots of people in the room probably know about. But the pseudo-random patent is Cirtec's property now, and so we built a chip around this. So here's what it is. It's 4 independent channels, so 8 electrodes. You can see on the picture, they're fully differential. We come in first with a multiplexer, so you can rearrange which electrode goes to which amplifier, but always in a fully differential fashion for this chip. We come into this low-noise amplifier high-pass filter combo, which is that chopper amp, into a programmable, first an anti-aliasing filter, then a programmable gain array, so you can cover a wide range of different signal sizes. Then we go into a delta sigma A to D converter, and we do the first round of digital filtering on chip. And then the output is a digital word that comes out, and so it's basically 4 known good digital words representing every sample at the input. And then down at the bottom, there's just overhead stuff, control registers to program all those variable, like all the specs say, all the variable filters, the variable gain, all those are programmable via SPI. So some of the key things we think, it might be the industry's best once it's done. If you take the combination of the noise floor and the power consumption and the size. So the key there is 350 nanovolts RMS noise with really low power consumption. So that's it. This is just getting into the details. Here's the spec sheet. There's a picture of the layout, so it's real. The design is complete. We do not have them in hand yet. They're coming real soon, but within the next couple months, we should be able to sample these and hopefully help you guys all do whatever you're working on next. And I think that's it. Oh, just one more loop is why are we doing this? It enables closed loop modulation, right? So we had a bunch of stimulator circuits. Now why don't we do some sensing circuits? And then the next step, of course, is close the loop. And this is just an example where the yellow is our ASSP stuff. And for the most part, the blue boxes are standard products, so you don't have to reinvent them. And then in this scenario, the only true custom component in order to make a set of closed loop hardware would be the custom coil. That's usually has to be customized device by device. But the rest of it, we hope we're just way up the curve in able to be able to make these things quickly. That's the idea. I think that's it. Other than thank you for reminding me, we're hiring too. so give us a hit anytime you like. Thank you.
Thank you so much, Andy. Okay. Remember, my grad students, not until you finish. No, I'm just kidding. Okay, so the next speaker will be Mark Hjelle, who is coming over here, and is Director of Global Technology Platform Development for Heraeus Medical Components. He is a University of Minnesota graduate in chemical engineering and chemistry. He has been working in the medical device field for over 33 years, and a breakdown of that, 23 years at Medtronic, 3 years at St. Jude Medical, and 7 years at Heraeus. He's also IEM Industrial Fellows member and a great guy who's been helping a lot with many of our programs and events here at the university. So looking forward to your talk titled, there. Oh, it's on the screen. Oh, there we go. Amplicoat, a solution for neuromodulation systems. Okay, got it. Perfect.
All right, given that I fell off the stage and the computer fell down a little bit, I'm just going to stand right here, and hopefully gravity is kind to me for the next few minutes. All right. So it's an honor to be able to present in front of all of you on behalf of Heraeus Medical Components. Heraeus is a tier 1 supplier. We are a contract design manufacturing organization, very similar to Cirtec, little different areas of emphasis as we go through. So we all are competitors in the room at certain points, so we all respect that and enjoy that, and it makes us all better. So that's a good thing. My co-presenter is Dr. Suman Narayan. She's based in Germany, where our corporate headquarters are. Our divisional headquarters are here, where I am. Suman is my partner in crime. She's the brains behind all this. We do all the work over there, and she puts up with me. Leonard Stoika has been focusing on the acute applications for which we use Amplicoat, and then Andreas Reisinger is their boss and my peer. So, neuromodulation market. Right now, all the electrodes are uncoated due to their design, materials, manufacturing processes that are used for their products today. So, CRM, in perspective, has been using coated electrodes to improve their sensing performance and stimulation performance since the early 1980s. So, there's a difference, and there's an opportunity. From a market driver standpoint, we hear that people want to have improved sensing. They want to have accurate diagnostics. They want to increase stimulation capacities or capabilities, and they want to increase battery longevity or increase the time interval between charges. We believe that Amplicoat can provide a solution for reliable and robust coating for these market drivers within the confine of your current product design. What that means is you do not have to change your lead design to use Amplicoat. Amplicoat was developed by Biosensing, which came out of the University of Michigan, which I think half of the PhD people here went to school with Jeff Hendricks, and I think you all drank beer at University of Michigan football games, but we won't talk about that, but there are pictures. And so we purchased them in September of 2017. We have acute applications that are approved for the FDA and CE mark for EP diagnostic catheters, those bird cage catheters that are used for diagnostics for EP. For chronic use, we've been working hard the last couple of years. Suman and myself and her team have been working to develop that, and we have a couple of customers that we're gearing up for co-sponsored chronic animal studies, and then hopefully next step for commercialization. So the EDOT to PDOT oxidative polymerization reaction is shown right here, and it's a pretty common item. PDOT's been around for a long time, but what Jeff was able to do, what Biosensing was able to do, is to change the additives to make it biostable. The process that we have for chronic applications is Amplicoat Plus, and that Plus is surface preparation. What we do is we laser structure the electrode to improve its adhesion properties. Then from there, then we clean it, then we do the electrochemical deposition of the Amplicoat, we do a UV curing process, and then we can optimize the coating for your performance. Basically, the thicker the coating, the more charge you can inject, the more you can store. The thinner the coating, the better the sensing. There's some parameters on that, but that's basically how it works. So this is a system change. It's a change to the lead, but it's an actual system change to tune the electrode to the performance that you want to have with your IPG or your INS. The Amplicoat process is really quite simple because there's no masking involved. It's electrode deposition. It's good old-fashioned plating. It goes where the electrode is. That's it. So this is a simple PCB flexible tape, and so you can see the gold side that's uncoated, and the dark side, it's more of a dark blue, but it looks blackish. And basically, it's just you charge one end, and you give it the right pulse, and you drop it in there, and you've got a coated electrode. One of the biggest benefits is high-definition sensing. So this is published literature showing the comparison between PDOT, or Amplicoat, compared to iridium oxide, which is a standard coating that's used in CRM. And it's a 50% reduction in polarization. That's significant. Increased stimulation capabilities. You compare the two waveforms there, and it's a 10x increase in the charge injection capacity for a given pulse, which is significant. From a charge storage capacity standpoint, you can see about a 15 to 20x improvement for a given electrode. Battery life. Battery life is what is gets a lot of people excited about, because where you really see the improvement is in the low frequency ranges that a lot of people want to operate in for closed loop situations or closed loop scenarios. And so you can see the tremendous difference, almost 2 orders of magnitude difference between an uncoated electrode and a coated electrode. And this is something that, again, is published when we did some work here with Matt Johnson, did some numerical modeling, presented that at NANS 2 years ago. The other thing, too, is I don't know what you've ever seen from a supplier standpoint, but I don't think anybody comes to you and says, I want something bigger. They always want something smaller, and you're like, really? And but this really promotes smaller electrodes. So if you start looking at really smaller arrays, trying to get smaller, you've got compensation and trade-offs you need to make from a system perspective, not just the lead, but from a system perspective. And this really does promote smaller electrodes because it deals with the impedance in a better manner. When you start thinking about chronic devices and coatings, you start thinking about reliability, you start thinking about this device is going to be in there for 20, 30 years, and what's the adhesion going to be like? How do you measure adhesion? It's not really an exact science. So what we tried to do is develop a test method. So this works for both leads and for flex PCB tapes. This happens to be a flex PCB tape. We have just a normal load applied, and we can do 20, 40, 60, 80, 100, 20 grand increments. And we go back and forth and slide and scrape across that tape or across the lead to see if the coating will fall off. And it's a saturated condition both for the Q-tip and for the sample. And so we feel really comfortable with any substrate getting around 200 grams of force applied. This is a summation of all the kind of jungle testing that we've been doing pulse stimulation, thermal shock testing, prolonged CV stimulations where we would work within the water window, accelerated aging with abrasion tests, and then we also did hydrogen peroxide reactive oxidation aging. And you can see that if you look at the bare electrode to the laser structured only, you've got a 10 order of magnitude improvement. You have another order of magnitude improvement if you go down to Amplicoat plus laser structuring. And then the last piece is biocompatibility. This is not all the tests for 10993, but it's a number of the tests. And we have very good performance in that. Obviously, there's probably some more tests that would need to be done when you're introducing a new material, but our purpose was to burn down the risk so that a customer would feel comfortable, and we do have customers comfortable wanting to go forward with this on this. So that's it. Just one more last slide, but we're done. Thank you.
Thank you, Mark. So moving along here, we have the next speaker is Dr. Gaynor Foster, and she directs, Director of Research Product Management and Scientific Affairs at Maxim EGI, directs Research Product Management and Scientific Affairs. They're the global neurotechnology leader that supports research labs, clinics, hospitals, and universities that focus on mental health, brain disorders, and cognitive neuroscience. She received her PhD at Warwick University in the UK, where she was an early adopter of HD EEG research technology. Really cool. She went on to leadership roles with the pioneers of HD EEG electrical geodesics Incorporated, EGI, now Maxim EGI. As a scientist building tools for scientists, Dr. Foster is inspired to share imaging and stimulation technologies rooted in research. So looking forward to your talk. Thank you. I was trying to look for the title here, but it didn't have one.
No problem. Thank you very much. It's really lovely to be here with you today, and especially as Minnesota is now my home. I've been living here a couple of months now, so lovely to be talking to you from my home turf. As the introduction said, I lead up it looks like it's not quite showing the title slide, but that's fine. I lead up the research product management team here in Plymouth, and obviously while I'm curating that portfolio, one of the main things I'm thinking about is innovation. And what does innovation mean? Well, we all want to be associated with it, whether it's through the products that we're developing or whether it's through the applications that you're using them for. So practically, though, innovation can only happen when we combine the right technology with the people who want to use it. As a scientist and an early adopter of HD EEG, I understand the challenge of adapting to a new technology. And it was only through close collaborations with my colleagues that we were able to take what felt like an unknown commodity and taking it and actualizing it to become an essential tool for understanding the dynamics of the brain. My breakthrough moment was realizing that looking at EEG data in a different way unveiled the true potential of the information that was contained within it. It was pivotal in my understanding that technology shouldn't be pitted against one another, but rather instead we need to combine modalities to ask the right questions with the right tool. And I strongly believe that evolution of neuroimaging is exactly where we are with neuromodulation today. I feel like we're on the precipice of a neurotechnological revolution where our collective goal is to bring about fundamental change and improve patient outcomes. And we can facilitate this by combining the right technological advances to provide you with the tools that you need to take the area forward. We know that electrophysiological data has exquisite temporal resolution, and so it allows us to identify with millisecond precision when things are happening in the brain. Its utility within source estimation, however, is confounded by the fact that volume conduction limits its spatial resolution, inferring that we may not be able to see when something happens, that while we can see when something happens, we may not know exactly where it does. Stefania Conti and John Richards have identified that it's critical to know exactly where sensors are during data acquisition, and then use head models based upon the individual's anatomy to increase the quality of the source analysis results from EEG data, therefore being able to also maximize that spatial information. This reinforces the application of the reciprocity theorem in targeted electrical stimulation, whereby if we know the scalp distribution of an electrical potential, which we show through doing source estimation, it can be linked to that specific cortical target, and by stimulating that same dipole on the scalp, we can innervate that cortical target also. Within our portfolio, we provide researchers with the tools that allow them to identify where sensors are located, to go on to create those individual sensor head models, and then also plan and deliver electrical stimulation in this targeted fashion. By supporting you in this way, we pave the way for that fundamental research to be done that can not only quantify the impact of targeting neuromodulation, but also look to ways in which it can be optimized. So, for example, is there a way in which we can use this modulating effect of electrical stimulation to facilitate the stimulation that we get from magnetic stimulation? Or can we optimize the magnetic stimulation by coupling it to the dominant frequency of the cortex? Another practical expression of how circumstances can also lead to innovation is how MagStim EGI came to be. When EGI's portfolio was purchased by MagStim in 2020, I think we're allowed to mention that year now, I'm not sure. We were able to bring together the leaders in dense array EEG and electrical stimulation with the leaders in TMS technology. And so this has allowed us to fully explore how to get the best potential out of both those technologies together. Marrying together EEG and TMS in a clean workflow couldn't be more important at the moment. Fowler et al., under the guidance of one of the true leaders of TMS, Mark George, have demonstrated phase entrainment through repetitive TMS. Taking EEG data and reacting to the dominant frequency characteristics in real time not only allows us to optimize to the dominant frequency of the individual, but also then can allow us to phase lock the treatment into that frequency. Findings like this are in harmony with our goals of using the EEG signal to both target and optimize neuromodulation protocols. But it does lead us to the next question. Have we found the most effective treatment frequency? We're now comfortable with the knowledge that the frequency of pulses delivered changes the consequence of neuromodulation. Providing our researchers with a stimulator that allows you to do more than you think you need to do, allows people to explore the pattern of stimulation that's used and identify those frequencies that go on to either inhibit or activate the cortex. And so rapid TMS protocols were developed. By understanding the optimal biological environment required to support continued excitation, we saw the introduction of accelerated protocols. And not only are we seeing the optimization of those protocols now, but we're seeing more treatment opportunities. As we see here, where Wu and their colleagues demonstrated that an accelerated rTMS protocol provided symptom relief for Alzheimer's patients. A beautiful consequence of these accelerated treatment protocols is that positive impact that we have on a patient's overall experience. Not only is there the chance for more immediate improvement, but across the population, we get to have the equity of healthcare. We have the potential to treat many more individuals in a more effective way. So alongside this, we're also improving on the infrastructure to support the much more data-driven approaches we are taking and optimize the treatment and efficiency with which this can be deployed across populations. Thanks for giving me this opportunity to share these thoughts with you. We take our obligation to provide you with devices that do more, and more importantly, more than you think you need now, really seriously. And we have a growing scientific liaison team that are skilled in all aspects of imaging and stimulation, so we can be here to guide you through these advances. Thank you.
And welcome to our warm and sunny Minnesota. So our final speaker is Merrick Gorley. Is the manager for DBS Science and Engineering at Boston Scientific Neuromodulation. He trained as a biomedical engineer at Georgia Tech, Emory, and the Johns Hopkins University. He has spent the past 12 years specializing in functional neuro, most recently overseeing the launch of image-guided programming for DBS, and actually the title, Image-Guided Programming and Intuitive Adaption to Managing DBS Therapy. So for the last speaker, thank you.
Good afternoon, everyone. Thank you for having me. My name is Merrick Gorley, and I'm a manager of DBS Science and Engineering at Boston Scientific. We're obviously very proud to be here, and I love just the spirit of collaboration and innovation that's happened over the last couple of days. We are especially appreciative to the University of Minnesota. The clinical team here, led by Dr. Vitek, was the primary author on our pivotal study. Sorry. Did it switch off?
Is that better? Yeah, it is. Whew, there we go. So just a little bit of background very quickly about myself. Trained as a biomedical engineer. I've been in the functional neuro space for about 12 years. So as I decided to start to work for a company in industry, I really wanted to work for a company that led with innovation, put a lot of money into engineering and R&D. So while I'm here today to talk to you about our latest development in the image-guided programming portfolio, which is StimView-XT, hot off the press, got FDA approved last week, I really wanted to give you, especially you that are kind of also maybe thinking about what you're going to do next, kind of a look back at what we've done so far from an engineering perspective in the field of DBS. So challenges that have kind of historically existed in DBS that we have since, we think solved. First, there were frequent IPG replacements, which led to increased infection rates in these patients with primary cell devices. So we introduced the industry-first 15-year rechargeable, which had the smallest form factor of anything else on the market, and really had patient peripherals that were designed for a movement disorder patient that kind of broke through the barriers that prevented patients from having rechargeable devices historically. Then we kind of looked at what you could do with those devices. And we found that there was really poor stimulation control with single-source IPGs and voltage-controlled IPGs. So we introduced MICC, which was a dedicated power source to each contact on the lead, which essentially allows us to put stimulation anywhere along that active span that the programmer thinks you need to and that the anatomy would dictate. Then, of course, we wanted to steer away from side effects because not every DBS lead can be perfectly placed. So we introduced the world's first directional DBS system. Then from there, we started to look at patients that have had DBS for quite a while. And we noticed with these legacy systems, compared to what was new on the market, these patients were really falling behind in terms of therapy. So we introduced an adapter that would allow you to hook up our IPG to legacy DBS leads and give the programmer and that patient the benefits that all new patients have, things like hypnotic stimulation, MICC, and semi-bipolar. And that's recently got cleared by the FDA as an MRI conditional swap. In fact, there's a study that was just published in April out of the Stanford group that showed patients that had these upgrades, 68% of that group had either an improvement in their therapy or a reduction in side effects, and 91% found their best setting was on some sort of fractionalized level. So there's no question that MICC is really changing the game and what you can do with stimulation. So what's left? Well, we at Boston Scientific now think that programming a DBS device is still really a trial and error approach. So that's where StimUXT comes in. We've now got patient-specific anatomy on the programmer that gives you a great idea of what level of the lead is going to be therapeutic, and when you encounter a side effect, how you might turn the stimulation or steer the stimulation to avoid that side effect. And as far as technologies that attempt to tell you how to stimulate, we think this is a really elegant solution. It's available for every patient, and the information that you're seeing is really intuitive, and you can act on it clinically very quickly. So what's behind those pretty pictures that we're showing? Well, BrainLab and Boston Scientific over the past 15 years have developed a multimodal voxel-by-voxel tissue classification system that basically takes the pre-op DBS scans that were done, then based on the structure that it wants to segment, uses the best scan for the job. So if we're segmenting the STN, if a T2 is available, we'll preferentially weight that modality to do that segmentation. And you wind up with essentially a 3D copy of your patient's anatomy on the programmer. This is independently validated by reviewers each time, so we make sure whatever we're putting out, at least yearly, is at least as good as the previous iteration. And now it's been kind of validated in the literature, both in a surgical sense, comparing it to MER, and then in the clinical sense, actually programming patients based off of it. So I'd like to very quickly delve into two of those studies that are getting fairly recent. So this one is out of the European group with Dr. Reich, Dr. Steigerwald, and Dr. Volkmann. And what they did was they basically compared image-guided programming or anatomy-based programming to the classic way that we would program a device, which is monopolar review. And what they found was that the motor outcome between the two methods was statistically the same. The big difference is that using the images in anatomy resulted in a programming time that was at least 50% faster. And that's what we're seeing across the board. Institutions that are using this are getting their patients to a good stasis faster and in fewer programming sessions overall. And you can imagine what that level of efficiency kind of adds into your clinic. Here's a paper from some United States groups that essentially showed the same thing. 2/3 of their patients were programmed in under 30 minutes with an average overall programming time of 34 minutes. So let's actually take a look at how this works. Here's an example of a lead that is a tad lateral and maybe a little bit deep. So with our system, utilizing MICC, we can turn on stimulation, scroll the VTA up the lead, kind of stop in between levels and evaluate the patient. But now you've got the ability to look at portions of the anatomy that you didn't have before when you had to jump from level to level with the contacts or single segment activation. So let's say we got to this point on level 3 in a dorsolateral STN, and because we're lateral, we started to have some sort of capsular side effect. Well, the anatomy does a great job of dictating how we can steer that VTA away from side effect and into the dorsolateral STN. And we're not locked into this kind of static view. You can rotate this in 3D and look at the programmer and say, okay, what is my next step? Make it intuitive and an informed guess as to what your next programming strategy would be. And one of those may be the use of semi-bipolar stimulation. And what that is basically using just a little bit of a hypnotic current, taking it off of the IPG and putting it onto the contact that's facing the side effect. And what that does is it actually creates a buffer of cathodic activation of that side effect. And then on top of that, by lowering the pulse width, we can really dial in the VTA in the dorsolateral STN. Now there's some data to this. The Record Group recently put out a paper in 14 of their patients that had directional cathodic stimulation but still had tight capsular side effects. They employed this method, and the therapeutic window widened in those patients. And in 12 of the 14, the side effect that those patients were experiencing basically melted away with this method. And this is only possible with a Boston Scientific device. So I'll leave you with this. It's a kind of an innovation form. I don't want to leave you with just what we have on the market. I want to let you know what's coming down the pike. So, what if in the future of DBS, we gave you a 4-channel IPG and expanded directionality, not just for use in movement disorders, but also for use in other indications like psychiatric indications? What if we gave you access to normalized heat maps that both on a patient-specific and a symptom-specific level guided you on how to steer your simulation? What if we changed the way that DBS was actually delivered, changing the waveforms in hopes to maybe prevent habituation of the thalamus or be a little more selective on the neural pathways that we're activating? And what if we fully closed the loop, not just for sensing, but use information from wearables and image-based techniques to essentially, in the clinic, give you an idea of some semi-automatic programming techniques, and then also, as the patient lives their life, kind of adapt the stimulation as needed. So I know that's a whirlwind Boston Scientific tour, but if you agree that we've done quite a bit in the field of DBS in just the 4 years that we've been on the market, just imagine what we're going to do in the next 4. Thank you.
Thank you so much, Merrick. Great talk.